Okay, so let's start talking a little bit about, some of you might dread this, but the Hardy-Weinberg equilibrium. I think once you understand it, it's not going to be, you know, too difficult to grasp. First thing we need to talk about is what is Hardy-Weinberg equilibrium? And it describes a population which is not evolving. So I'll write that over here. Population that is not population that is not evolving. Now, I want you to write down, what does a population look like that is not evolving? Go ahead and pause the video and try and write down what a population looks like that is not evolving. Okay, if you are back, let's go ahead and go through this. You might have been a little bit stuck and thought, how do I know if a population is not evolving? Well, you can think again of your 5 fingers of evolution and reverse them. So, little finger, 1. A population that is evolving a lot of times is due to a small population size. So this is typically going to be in a large population. A non-evolving population. Number 2, you'll remember that ring finger, right, represents non-random mating. So a Hardy-Weinberg population, which is not evolving, should have random mating. Random mating. Okay. Number 3, should have no new alleles introduced via mutation, because of course populations with a high mutation rate are going to be evolving much more rapidly. So these populations would have no mutation or extremely low mutation at a certain allele, but we would assume no mutation. Right? No mutation. Okay. 4th, no gene flow. Remember, populations that are evolving have immigrants and emigrants, right? Individuals coming in and out of the population. This is not occurring in non-evolving populations. Okay. And the 5th one, probably the most important one, no natural selection occurring. Okay, so I'm not really going to do a lot of calculating in this first Hardy-Weinberg video. I want you to understand the basics of things. So the first thing I want to remind you of is an allele. And I'm going to draw a big, thick line. Pretend this line is super thick and it cannot be crossed, because I don't want you guys confusing these two things. And genotypes. Genotypes. So alleles are things you've seen before. They are these single letters that represent the certain mode of genes. So for instance, we could say, big A represents, is a dominant allele, which codes for maybe brown eyes or something, right? this is kind of the classic example. And little a over here codes for blue eyes, right? We're going to assume that this is just a single gene trait, and big A codes for brown eyes, little a codes for blue eyes. Each of these is an allele. Now, what genotypes does this give rise to? Well, you probably remember this. You get one allele from mom and one from dad. Big A. So these are the different combinations you could, in theory, get from these people. I'm going to draw another line over here so you can see phenotypes. Phenotypes, of course, are the result of genotypes. They give rise to phenotypes, or how a gene is expressed. I'll just put expression of a trait, right? So we know that big A codes for brown eyes, and it is big A, so it is dominant, which means that the genotypes which will code for brown eyes are big A, and big A, little a. And the genes that code for blue eyes, the genotypes that code for blue eyes, is little a. Now this is probably all very basic to you. This is probably stuff you remember, but I do want you to recall how this basic system works, because when you're working a Hardy-Weinberg problem, this is what you're going to have to think of. So we're going to use different variables in Hardy-Weinberg to represent these alleles. Little p and little q. Little p is going to represent the dominant allele frequency. So I'm just going to put frequency of big A. Now, it doesn't matter which one you use for dominant or recessive according to your formula sheet, but I'm going to stick with this convention because I think it'll keep us all on the same track. Q represents the frequency of little a in a population. When I say frequency, that is like a percentage, right? Okay, let's move on to genotypes. Big A is going to be represented by P squared, or P times P, which is going to be the frequency of big A genotype. Keep these separate. Definitely keep these separate. Do not cross this line right there. Okay, 2PQ. Oh, let me go back. It is going to represent the frequency of heterozygotes, or big A, little a. And finally, Q squared is going to represent the frequency of little a. Now, you might ask yourself, we've got some variables, P, Q. These are going to give rise to expected genotype frequencies of P squared, 2PQ, and Q squared, the frequencies of all these different genotypes. But what is the purpose of this Hardy-Weinberg equilibrium? Why do we want to know what a population looks like when it's not evolving? And the reason we want to know that is because it gives us a valuable null hypothesis. I.e., if we could actually do a chi-square of this, right? We could do a chi-square if we could find out what values we would expect in a population to be certain genotypes. If these values are not close enough, then we'll know that our null hypothesis is wrong and a population is evolving. So this is the value of the Hardy-Weinberg equilibrium. Now I want to show you kind of a square that kind of illustrates how this Hardy-Weinberg works, and hopefully it will illuminate things for you. I'm going to pause the video, pull it up, and then we will play with it. Okay, so this is courtesy of a Matt Thomas online. I want to give credit where credit's due because I did not program this myself. He did a great job of it. It was over at a website, a geometry website, which I'll list on Schoology for you guys. But if you look at the top, you'll see our variables here. P, and little q, and then little p and little q over here on this side. So you'll notice inside, if you multiply, you're going to get the area of this square, really. 0.5 times 0.5 gives you 0.25. 0.5 times 0.5 gives you 0.25. All of these squares are of equal size, so you can kind of see where it comes from. Of course, P plus Q will always equal 1, or 100%. P squared plus, again, there's two of these PQs, plus 2PQ, plus Q squared also equals 1. And I think if you did the addition on that, you would come to that conclusion, right? P plus Q, 0.5 plus 0.5 is equal to 1. And 0.25 plus 0.25 plus 0.25 plus 0.25 is also equal to 1. Now let's see what happens when we make P very large. What happens to the area of these squares, right? I'm going to bring P up to 0.9, very, very large. You'll notice that the area of P squared gets very, very big, right? 0.9 times 0.9 is 0.81. Keep in mind, what this means, and this is the key takeaway from this, is that if 90% of the alleles in your population, remember alleles are the single version of genes, or the single modes on a gene. If 90% of the alleles, or 0.9, frequency of 0.9, are dominant, then that means that 81% of your individuals, or 0.81 frequency of your individuals in your population, will have the homozygous dominant phenotype, meaning big A, right? Conversely, if you look at Q over here, it's fairly small, 0.1 and 0.1. 0.1 times 0.1 is 0.01. This means that about 1% of your population would be homozygous recessive. This is what you would expect were evolution not occurring. So this is how we, in the next video, how I'm going to show you, we're going to calculate this. Let me flip this back the other way, just to show you that sometimes the recessive alleles in a population can be just as dominant. Just because they're recessive doesn't mean that they can't dominate a population. If you look here, Q squared is at 0.81, right? 0.9 times 0.9 is 0.81. This means that most of your individuals would be homozygous recessive, right? If you look at PQ up here, your heterozygotes, this area represents how much of your population would be heterozygous. 0.09 plus 0.09 give you 0.18. And of course, you have this small percentage here, this 1% which is homozygous dominant. So this is something fun to play around with to kind of visualize what we're going to be looking at when we're looking at these percentages, because this is going to give us ultimately our null hypothesis. This is the hypothesis we can test against to see if our population is evolving. Okay, this will continue in the next video.